The last line に僕らは歩き慣れてきた日々も問うた夢は安泰な暮らしだが刺激不足上にダラダラ照らす暗闇僕らの歩き慣れていた道はどこだ時はたまにシャッだがぬくもりに包まれたらほんのりがあったシルベの方へ思い出すは優しいメロディー永遠はないなどないなどよそれもまたいいなと笑ってみる輝けばいつかは光も絶え僕らは命の火が消えるその日まで歩いてゆけ音が出る玩具も痛みを飛ばすのも全部僕にとっての宝物永遠はないだとないだという僕らは命の火が消えるその日まで歩いてゆけ
So, the topic we have chosen is all about enzyme action. But first, to understand how enzymes work, we must first know what an enzyme is. So, basically, enzymes are proteins that are made up of amino acids. Other than that, they are known as biological catalysts. These biological catalysts are the molecules that are capable for speeding up certain chemical reactions such as in photosynthesis, respiration, digestion, and protein synthesis. In order to do this, these biological catalysts lower the activation energy that are required for that certain chemical reaction. Because one thing to note here is that the rate of a chemical reaction can be increased only if the activation energy required is lowered, which is what enzymes and or other biological catalysts do. Here in our model is one right here as what we have introduced a while ago. This green clay with a unique shape represents the enzyme, while this red one right here is the clay that represents the substrate. This enzyme and substrate are always moving around, and at some point, collision theory occurs. In collision theory, the enzyme and the substrate bumps into each other at the right speed and orientation, and with this, the substrate can now bind into the binding site of the enzyme. So the simple analogy for an enzyme action is the lock and key model. As we all know, a lock can only be opened if and only if a key with a matching shape can fit into it. Now, with a similar context to that, using our model here, this enzyme will only allow a substrate into it only if its shape can fit into the binding site of the enzyme. And when this happens, it creates a covalent bond between them. And after the binding of the substrate and an enzyme, there are three ways that the substrate can be catalyzed. First is when an enzyme breaks down a substrate into smaller molecules. The second one is where it can do the other way around, which is to bind smaller substrates to become a single and new substrate. And the third one is where the enzyme can simply change a substrate into another new substrate. Also, when a substrate binds into an enzyme, they create what is called the enzyme substrate complex, which gives rise to the end product. So once the reaction is done, the enzyme releases itself from the substrate. The enzyme remains unchanged, and it then starts to move around, waiting for another substrate to bind to it. Now, moving on, we now go to the main parts of the enzyme, the binding sites. So there are two binding sites of an enzyme. The first one is the active site. The active site is the region of an enzyme where substrate molecules bind can undergo a chemical reaction. This site consists of amino acid residues that form temporary bonds with the substrate and also residues that catalyze a reaction of that substrate. And as we have discussed, these substrates can be catalyzed into three different ways. However, there is another molecule that can bind to the active site of an enzyme besides a substrate. That is what we call the competitive inhibitors. So, competitive inhibitors and non-competitive inhibitors are the ones that cause the process called inhibition in an enzyme. Note that when a substrate binds into an enzyme, the process that can happen is called reaction, while when an inhibitor binds to the enzyme, the process that can happen is called inhibition. So, competitive inhibitors are the molecules that competes against the substrate only to bind to the active site of the enzyme, hence the name competitive inhibitor. When a competitive inhibitor binds to the active site before a substrate, this occurrence prevents the supposed substrate to bind to the enzyme. Thus, a reaction will not happen at all. One of the reasons why competitive inhibitors are blocking the active sites of an enzyme is to prevent the formation of undesirable products. To visualize this, let's take a look at an example. One of the real-life example of competitive inhibitor is penicillin. Penicillin is an example of competitive inhibitor because it is capable of blocking the active site of an enzyme. So, when this happens, the bacteria that was intended to bind to that enzyme in order to construct their cell has been blocked off. Thus, the formulation of the certain bacteria was prevented. And that's how competitive inhibitors work. Now, we move to the allosteric site. So, aside from the active site, we have the allosteric site. Allosteric site is a region of an enzyme where the activity of an enzyme can be significantly affected. The molecules that bind in this, the allosteric site, are called the non-competitive inhibitors, where non-competitive inhibitors takes place in order to change the structure of an enzyme, preventing the enzyme from catalyzing the reaction efficiently. One of the goals of non-competitive inhibitors is also to prevent undesirable products, which is quite the same with the competitive inhibitors. Let's take a look at an example. One of the real-life examples for non-competitive inhibitor is aspirin. When aspirin binds to the allosteric site of an enzyme, it prevents a certain product called prostaglandin from being produced. And when this happens, it greatly benefits the human body. For example, it reduces pain, fever, inflammation, blood clotting, and others. So, those are the basic ideas and concepts regarding enzyme action. So, to summarize the ideas, enzyme is a biological catalyst that speeds up chemical reaction which involves substrate. And there are three different ways for an enzyme to catalyze the substrate. First is to break down the substrate into smaller ones. Second is to combine two substrates to form a single substrate. And the third one is the changing of the substrate itself into a new one. Competitive and non-competitive inhibitors, on the other hand, are the ones that are responsible in affecting the activity of an enzyme. That is all. Thank you.
The last line に僕らは歩き慣れてきた日々も問うた夢は安泰な暮らしだが刺激不足上にダラダラ照らすは闇僕らの歩き慣れていた道はどこだ時はたまにじゃったがぬくもりに包まれたら本村があったシルベの方へ思い出 スは優しいメロディー永遠はないなどないなどよそれもまたいいなと笑ってみる輝けばいつかは光も絶え僕らは命の火が消えるその日まで歩いていて音が出る玩具も痛みを飛ばすものも全部僕にとっての宝物